But let me now introduce Kristina Gessel, who will tell us about a virtual unwrapping. This is part of the EDUCE project, which we'll hear much more about on November 18th. We've scheduled a full hour about EDUCE, and so this is a warm-up for that. And so, Kristina, it's all yours.
Great, thank you, Dr. Finkel. Let me share my screen with you. So hopefully you can see my slides. So, hi everyone, I'm Kristina, and I'm a PhD student working in the EDUCE lab here at UK. Thank you for the intro, Dr. Finkel. And the EDUCE lab applies custom software to CT data to read undamaged manuscripts without having to physically open them. A major part of the software pipeline is segmentation, which we will mostly be talking about today. Today I'm going to present a new approach to segmentation, and then I'm going to show some results that we've obtained by applying that new approach to a real manuscript. First, before going into any technical details, I'm going to introduce the manuscript. This is MSM 910. This is a book that was acquired by the Morgan Library, which is located in New York City, and they acquired it in 1962. M910 is a 6th century Coptic manuscript, and it's one of the earliest known copies of Acts of the Apostles in any language. In total, M910 has 102 pages, and we suspect that there's writing on both sides of most of these pages. The pages are extremely thin and also brittle due to some possible fire and water damage, which you might be able to see some evidence of damage on this photograph, as well as just its age. So it's impossible to open this manuscript physically. The text is written in iron gall ink, which we know generally shows up well in computed tomography scans, since the iron in the ink is denser than the page itself. So this book seemed to be a promising candidate for the current virtual unwrapping pipeline, and it was scanned at the end of 2017. M910 was scanned with a micro CT scanner to obtain a few different scans of the interior, and this is one of those scans. This is a 35 micron scan where you can see an entire cross section of the manuscript. So just ignore the gray circle for right now. We will get there in a second, but just taking a look at this cross section, you may be able to see a lot of damage. For example, here there's a hole in the manuscript. I don't think anyone knows for sure how that hole occurred, but I think it's possible that perhaps a cinder was dropped into the book while it was open or something and burned these pages. You may notice that there is a more dense area here. This is where a couple of pages are kind of glued together, which may also be indicative of some kind of heat damage or some other kind of trauma to the book. The gray circle indicates the area of another higher resolution scan that was taken in an area that we suspected to have the most ink in view. And that smaller 16 micron scan is the one that we focused most of our efforts on in 2018, and it's the one that we're going to focus on today. As I start this video, you can kind of get a look inside the manuscript. The entire scan of this part of the manuscript is made up of more than 5,000 thin cross-sectional slices. This is a look inside the manuscript, starting closer to the binding of the book and traveling outwards towards the outer edge of the book. Note again that you can see more visible damage. You can still see the hole. You can see a lot of complicated the pages are in very complicated shapes over in this area. Some of the layers seem to still be glued together. You can see a few minerals scattered throughout the book. And if you look along some of these pages, you may be able to see kind of some faint light gray signal that may be iron gall ink. So those are probably some kind of ink signal, and that's just happening because the iron gall ink is more dense than the surrounding page. So those areas end up a little brighter. But of course, just looking at this kind of scan, we can't make much sense of those ink signals. So it's necessary to take the scan and process it a little more so we can put it into a form that we can actually see the writing like we normally would, as if we were just looking at the page. And that's where segmentation comes in. Segmentation extracts these individual layers from the tomography data. But segmentation isn't an easy task. As an undergrad, I actually worked on this project. with 3 other undergrads. And combined, we spent over 120 hours on segmentation, but at the end of that effort, we extracted only 26 layers from this smaller scan. And mostly, we concentrated in this area that you can see on the left side of the scan, highlighted in green. This image doesn't show all of the pages that we extracted, but I included it so you can kind of get a better idea of the scale of the pages we were working with at the time. The largest of these 26 layers took more than 15 hours of work to fully segment. And as I showed earlier, this scan is only a small part of the entire book. We didn't end up making much progress on the larger scan that I showed you. So we learned that a new approach was necessary to make the larger pages more tractable, given our constraints on time and people. And starting in 2019, I investigated a couple of ways to make segmentation of these scans more tractable as my master's project. I ended up with an approach that makes segmentation of those large layers very possible. And this is kind of a visual overview of that process. But ideally, we'd like to completely automate segmentation. It's a very laborious process, and it's not fun. And we were looking at machine learning techniques to do that, but we have a large obstacle in that we don't have any labeled training data that would help us. So any kind of supervised machine learning technique is just impossible at this time. So instead, I proposed a segmentation technique that requires no training data. All this algorithm requires is a set of user-provided seed points, which you can see on the left here. At this step, we estimate the thickness of the page at every single one of these seed points. And then we keep the median of all of these thickness estimates. Then the second step is performing basically a bounded version of just flood fill. So starting from each one of these seed points, we extend a mask from that point using a minimum grayscale intensity value that a user defines. So the darkest possible intensity that the user wants to mask. And then we determine whether a particular pixel is added to the mask or not with that intensity value. Each operation of flood fill from each of these seed points is limited to a radius that is the median thickness that we had computed in the earlier step. And no point can fill any pixels beyond its own radius. So as each one of these seed points creates a small mask, they end up overlapping to form a larger mask in the end, which is what you see here. But in many cases, the minimum intensity value that the user provides doesn't allow flood fill to mask the entire space where the page really is. In fact, in this mask, you can see a few dark spots in the center, just where those points were too dark to be masked. This is a problem later. So we need to fill those holes and gaps. And I do that by applying a closing operation. Here, I've visually divided it up into the dilation step and then the erosion step so that you can see that the dilation step grows the mask and removes the holes in the center, and it also closes some gaps between parts of the mask. And the erosion step shrinks the mask back down to about its original size, but now all of the holes and the gaps are filled. So once we have this mask, we can apply skeletonization to skeletonize the mask. And I chose to skeletonize by using a pretty straightforward thinning algorithm that thins the mask evenly. So as long as the mask is continuous, we're guaranteed to get a continuous skeleton in the end, and the skeleton is also centered inside the page, which is something that's really important for this particular manuscript. Because of its two-sided nature, there's ink on either side, so we need to be able to clearly separate the ink on either side. Now, the skeletonization technique can leave little spurs that do branch off the main skeleton, and we do a really simple pruning step to prune those the best that we can by just searching the curve for small branches and removing them. Some get missed in that step, but the end result still works out okay for us. So the scans in the manuscripts are sufficiently high enough resolution for us to see the ink, which means that the pages are also only moving very slightly between individual slices. So that means that we can use the skeleton that we created for a given slice as a set of seed points for the next slice. And as those are used as seed points for the next slice, the algorithm will recenter the new skeleton for the new slice. And this process can continue through the volume. There are some drawbacks with this algorithm. Sometimes the mask will leak into other pages, so it's not fully automated. A user does have to watch the algorithm and correct it when it begins to leak into other pages. That often happens when the pages touch or when they merge, like I pointed out earlier in the scan. But with the aid of a user, this algorithm is capable of segmenting the pages that we were unable to previously segment. The algorithm tends to be pretty stable, so it can often run for hundreds of slices without needing human intervention. And that's a big improvement over the previous segmentation approach, where we often couldn't segment more than 40 or 50 slices at a time on this particular data set, just due to the complex shape of the pages. It's also much faster than the previous approach. It segments more slices per second, which saves us a lot of time spent just waiting for the algorithm to complete. We are working right now on extracting the largest pages in the scan that I showed you earlier. And on average, we're extracting one of those pages in about 5 to 6 hours of work, which is a huge improvement over 15 hours for a smaller page. This algorithm alone is capable of segmenting most of the rest of the pages from the M910 manuscript, but as a user does that, it's also generating these masks that cover the page in most cases. These masks may need some cleaning due to its potential for leaking into other pages and things like that. But once the masks are cleaned, they may be able to be valuable training data for a supervised machine learning approach later on to fully automate this. So we get a skeleton for every single slice that we're interested in. And once segmentation is complete, we merge all of these skeletons to create a three-dimensional point cloud. We manually clean that point cloud and then mesh it. And once we have the mesh, then we can use the rest of the virtual unwrapping pipeline to get our final results. This is the best result that we've obtained so far. The ink signal came through really well in this page, well enough that it could be identified as Acts of the Apostles, chapter 10, verses 4 through 6. We're still learning a lot about this manuscript. And as we've been extracting these full pages, we've seen a lot of things that have surprised us. First of all, you may notice the ink signal is very strong in this region, and then it gets a little weaker as we move over in this region. And we aren't really sure why this is happening, but this trend is pretty consistent through the whole book. And it's one of the challenges that we need to overcome. And another thing is, for this particular page and some others, one side of the page comes through very strongly, and the other side isn't as easy to see. This has happened to some pages, but not all, and we're also investigating why that is. We're not sure if there's something maybe going on with the ink or the condition of either side of the page or what's happening there. This is another interesting example of a page that we found inside M910. Looking at the top, this has gone through the same process as the page I just showed you earlier. So looking at this, I'm not sure that this alone would yield much of an interesting translation or any kind of identification of this page, but this is a very interesting page for a different reason. The top image is what we get from just our regular texturing, like I said, which is enhancing the ink signal by starting from the center of the page and traveling outwards along the normals of the mesh towards the surface of one side or the other. And it's keeping the brightest intensity that it encounters as it travels along the normal. But the bottom image isn't starting from the center. Instead, this image is starting very close to the surface of one side of the page. And while here we don't see much bright ink signal, we do see these dark characters. And we suspect that these dark characters are evidence of the ink eroding away at the surface of the page over time. You can also kind of see hints of this in the top image where there are holes over top of where there was ink. So we think the ink is kind of eating away at the page and making it even thinner than it already is. So we've seen several clues of this as we've been segmenting more pages. But we suspect that these morphological cues are going to be really valuable in enhancing what ink signal is there, and even maybe finding some ink signal that has degraded too far to be detectable via CT through just density alone. The pages that I showed previously are from that higher resolution volume that I showed you. So they're focused at a specific part of the manuscript where the most ink should be. But this algorithm is capable of segmenting entire pages as well. And this page is an example of that. This is part of a choir of 6 pages that fell off the front of the manuscript. So they were scanned separately. For this page, I segmented 3,200 slices with the new algorithm, and that took about 4 hours to complete. And this one is quite interesting because you can see the entire shape of the page, and you can compare it to a photograph, which is really, really useful to see, you know, where we can improve and what we already have. So much of the ink signal is visible on the textured image on the left. You can see, especially in this region. But we do see some of the inconsistency that I mentioned earlier, especially in this area of that page. The ink signal just seems to get weaker as we get closer to the outer edge of the page, and also near the top where there appears to maybe been some kind of water damage or something poured on the manuscript. But this is a great example of where we can compare directly with photograph to help us further improve our ink enhancement algorithms. And it makes great training data for a machine learning approach to enhancing this ink signal. My final example of some results from M910 is a fragment that had fallen off of M910 at some point in the past. And we scanned this fragment at a very high resolution, 8 micron. And on the left here is just the raw mesh that I extracted with my segmentation algorithm. I cleaned the point cloud and then meshed it, and this is what I see. So you can see, just sampling straight from the CT data, a little bit of ink signal down here, but not too much. And that's actually good news for us at this point, because that means that there won't be a lot of ink signal to interfere when we get to directional texturing. We acquired photographs of both sides of this fragment for comparison, and I ran this mesh through our directional texturing algorithm, and you can see clearly that either side comes through quite clearly. The ink is not as strong as you might see on the photograph, and obviously there's some room for improvement in terms of that. But this is very exciting because the pages of M910 are so thin. So this is great news to see that we can, if we have a high enough resolution scan, we can isolate either side of the page without the other side interfering. So we made a lot of progress, but there's still a lot to do. First of all is to fully segment M910 as best we can. We've been working on this again since April, and since then we've extracted about 27 new pages from the volume that we worked on in 2018. So combined with the 2018 work, we've extracted about 55 of 94 pages in that particular scan. Of course, we want as many of those pages to be translatable as possible, which means we need to find ways to further enhance the ink signal. And we're working on that right now by attempting to use machine learning-based ink enhancement techniques. And this data set will be really helpful for that, because we will be able to have a lot of training data from the pages that we photographed of this manuscript. Another thing I want to investigate is actually using those generated masks as training data for a machine learning approach to doing the same thing. I'm sure if we develop a model on this, it may generalize well to other manuscripts in the future, even if we fully segment M910 without it. We also have some other data sets that have clear layer separation, but we just have never actually segmented them yet. So this algorithm shows potential perhaps for those data sets. So I'm very interested in trying them on those data sets as well to see how it does on those. And that's everything. So yeah. Any questions?
Let me ask a question. I gathered from what you said that given after the segmentation, you know, or you have an estimate of where a particular patch of a page is in 3-space. And then in order to find the ink on one side or the other, you start there and you move outward from on the normal from the page for a ways, looking to see if there's signal. Is that how it actually works?
Right. Yep.
Right. And so the question is, if you might be a little bit wrong after segmentation as to where the paper is, would it make sense to offset that algorithm by starting a little bit above or a little bit below in order perhaps to catch more ink?
Yes, absolutely, we could do that. Yeah, and that very well might be the little bit extra that we need to better isolate that. Yeah, that's a great idea.
Other questions? Because I've got another, but let me see if someone else wants to ask.
Rafi, I have a question.
Yeah.
Kristina, first of all, fantastic talk. You're doing some really interesting work. But let me ask a question about your pipeline. You said you ran these things through the pipeline, and then at the end you saw the results. And in some cases it seemed to work good on parts of the manuscript, and others it didn't. And you thought that perhaps by changing, well, obviously changing the algorithm, in some cases it may just be changing parameters, right. To the algorithm. And I guess the question is there any way of sort of exploring the space to see what might work computationally? In other words, could you run it through the pipeline, have an evaluation algorithm that evaluated how good the results were, and then change parameters and do it again, sort of zeroing in on a result? Is there any way to sort of, we looked at it and saw, boy, that doesn't look quite right. Is there a way to analyze it and understand that it's not quite right?
You know, I immediately can't think of a nice way to evaluate it because, you know, in many cases it is kind of a human, like it's kind of subjective, right? But I do agree with that. We are looking at running this through the pipeline with multiple different settings of the parameters, and it would be great to have an automated way of telling us, yes, this one is an improvement over this other set of parameters or not. I can't immediately think of a nice way to do that, but that's a great idea.
Yeah, thanks.
Yeah, thank you.
Anyone else?
I'll just add to that question, Kristina. I think like some of the machine learning stuff might be in a sense a way of doing that, where we replace part of the pipeline with a machine learning component that learns to reproduce this image. It only works right now on certain data sets where we have labeled data, but like Kristina showed an example earlier of one of these pages next to a photo of the page. Yeah, so in this case, you can train something instead of this pipeline. You can replace that component where it's trying to reproduce this image. So it sort of is learning inherently. It's not really there's no human in the loop, but that might sort of answer that.
Yeah, that's true. Thank you, Stephen. Yeah.
Yeah, if you know what you're going for, that would certainly provide a metric for you to be able to measure how close you are to it.
Right.
Yeah.
Do you have any control over the parameters of the micro CT scans? You're looking for a particular kind of signal, the iron-based ink. Are there any things you can tweak in order to maybe pick up the ink a little better?
Right. I will give you my best interpretation of what I understand. I was not present during the scan or behind any of the parameters, but yes, there are parameters you can tweak. And one of those parameters that I know certainly can influence how well the ink signal shows up is the energy level of the scan. And Stephen, you can correct me if I'm wrong, but I think the lower energy of the CT scan brings out the iron gall ink better in general. I'm not completely certain about that in particular, but yes, we can change parameters of the scan and potentially improve this too.
Yeah, that seems to be true from what we've seen. We would love to better, like more fully explore that. There's And that would be a case where it'd be really cool. I don't know how exactly it'd be done, but it'd be really cool to have it be sort of in the loop where at scan time you could tell how well the contrast is doing, and that could affect or directly influence the way the scan's actually performed.
Yeah,
We haven't fully explored that yet, but it seems like from what we've seen, when we scan these at lower voltages, the contrast is better for whatever reason.
So you could even scan it multiple times. How long does it take to do a physical scan? I assume it's in a library somewhere, you need special permission, special equipment, but how long does the actual scan take?
Stephen, do you know that one better than I do?
That varies, but typically with these, it's often hours, sometimes up to, and it depends on what resolution you want. So more time for more resolution because you usually have to let the, you need longer exposure times. So if we can, it depends a lot on the constraints of the actual document and the conservators and the whole environment. So there's a lot of constraints usually out of our control. So we usually make the scans as long as we're allowed to, but that can be as long as a day.
Right. Yeah, so you really can't make use of multiple scans at slightly different voltages, hoping that one of them will be better than others at a particular place within the document.
Right. We have been able to in some cases, and then, so then we have all this data that we haven't fully been able to sort of examine yet.
Have you made the data publicly available so other people around the world can try their hand at analyzing it?
We have not made this data set publicly available yet, but I am pretty sure that that is in the plan down the road. Since we're actively extracting stuff from this data set, we're really interested in seeing what we can see. But I do believe the plan is for this to be openly released so everyone can try their hand at it.
I have a question on that.
Sure.
So in our community, there's kind of an accepted standard that folks aren't expected to make their data available to everyone until they get a conference paper and a journal paper out of it. And then at that point, if you've had those papers and you haven't shared your data yet, people are saying, hey, where's the data? So I was just curious, is there some kind of a norm in your community?
I'm actually not sure about that one. Stephen, are you aware of any kind of norm for sharing data like that?
From what I have seen, it's something in between what you just described and people being a little bit more closed with their data sets.
Mm-hmm.
We are trying to be more open. Like Kristina mentioned, this one's not available. Some of our other ones are available that we've published already. Yeah, it tends to depend. And then there are a lot of works in this area that use scans of not synthetic, but sort of lab-made materials where the lab, in the lab, they will make a book like this and damage it themselves. So it's not such a high stakes artifact because it's just something made in the lab. Those tend to be people are more open with those for whatever, like there are lots of licensing and, whatever agreements with partner institutions and so on and so forth when you scan something like this that make it harder to make them open. But we're trying to change that.
Well, thank you very much for a fascinating talk, and we're all looking forward to hearing more about this EDUCE project in a few weeks.